So thanks to the Domino people for inviting me. Thanks for coming. Good morning. I'm going to spend the next 30 minutes talking about a couple important developments in the field of cancer therapy over the last decade. And both are sort of revolutionary developments and very exciting, but they came from different planets. And so biologists came to me and came to people like you to try and understand the connectivity and the exchange between these different types of therapy. I have 30 minutes. I'm going to go a bit faster and hopefully be thoughtful. So I work at Bristol Myers Squibb. It's a pharmaceutical company increasingly using biologics for therapy as opposed to pills in a bottle. It's an old company. It's been around since 1858. And notably, E.R. Squibb, whose name is still on my paychecks, was noted as being so unimpressed by the quality of medicines as a Navy doctor during the Mexican War that he pitched the unfit drugs overboard. So flash 160 years later, I think there are parallels in cancer therapy right now going on as we replace some older and generally very toxic chemotherapeutic drugs with some new medicines. I work in Redwood City, and I'm a bioinformatics scientist. Most of you probably have a feeling for what that is. It's sort of a slippery definition because we're a set of highly specialized generalists. We know more biology than a computer scientist, usually a lot more biology and genetics, but you can pick it up along the way. More computer science than a mathematician, more math than a biologist, and enough statistics to get into trouble and hopefully get out of trouble. And I work in a field called genomics, which I'm sure you've all heard of. The definition, it's a very broad field that tries to comprehensively study the genetics via the DNA, the transcription via the RNA, protein, et cetera, of an entire organism, of an entire species. The first human genome was sequenced in 2001. It actually took about 15 years to read about 3 billion letters. It took about $1 billion for one person. Now, in the meantime, the price of sequencing has dropped. On the right here, you see a Moore's Law line. And you can see that for several years, the price of sequencing dropped in accordance with Moore's Law. And then the bottom fell out. Sequencing became extremely inexpensive through advances in what we call next generation sequencing technology. And now we can sequence genomes in days to weeks and not decades. And the cost is like $1,000 or $2,000. So this is like the internet revolution for biology. And we can measure just about everything. We can take a human, take a blood sample, measure every genetic polymorphism in a person, every variation. We can measure the expression of every gene and its intensity through something called RNA-seq. We can do that for humans, for mice. We can do it for tumors. So we can measure every mutation, every genetic change that a tumor makes to become cancer. And we can also look at tumor RNA and compare it to normal RNA and find things that are different, that are aberrant. So the cancer community embraced genomics 10 or 15 years ago. And they've done remarkable work. And probably the pinnacle of that is something called the Cancer Genome Atlas. It's a very detailed molecular characterization of cancer using all of these techniques and more. So we have a database right now that anyone can access. You can download it today of about 11,000 patients across about 33 tumor types that have been characterized with 7 different genomic techniques. And so this is just a cartoon, an example of one way you can summarize this data. This is 200 or 300 patients in very, very thin columns in one tumor type. And these are the recurrent changes we see across that tumor type in terms of DNA mutations in the green, DNA amplifications in red, DNA deletions in those blue bars. And so we're looking for common themes here. One thing to point out sort of in the center is that protein TP53, which is also known as P53. It's an essential cell cycle control protein. And 73% of those tumors have knocked out TP53. And right below it, there's a gene called CDKN2A. You see that stretch of blue bars there, as well as mutations. So nearly half of the tumors in this cohort have either deleted the piece of DNA where CDKN2A was on, or mutated it to knock out its function. And why are tumors doing this? They're doing this for a couple different reasons. One is to either turn on cell growth pathways or to sabotage cell control pathways. And while they're doing this, they also have to evade the immune system. Any one of these mutations could be recognized as foreign by the immune system and attacked. Cancer therapists leapt on this data and changed the paradigm of cancer therapy. And instead of chemotherapeutic drugs, they wanted to target tumors. They wanted to find the driver mutations that lead to these proliferation dependencies, target them with drugs, and then sequence every patient, find the drivers, find the weak points, and treat them with the appropriate therapy. And so this is called the targeted therapy of cancer. It's also called precision medicine, and it can work remarkably well. So we have drugs to treat amplifications of DNA around a gene called HER2 in breast cancer, mutations in BRAF in melanoma, mutations in the epidermal growth factor receptor, EGFR, in lung cancer. And on the bottom here is a before and after picture of a patient. This was a patient treated with BRAF inhibitors, and unfortunately, all those black spots are tumors. This was a patient with a very high tumor burden, and a matter of weeks later, I think you can see on the right that most of those tumors are gone. So there are some fantastic results out of this precision medicine. Unfortunately, they do not always last forever, and many patients relapse. So the targeted cancer folks were patting themselves on the back, as they should, maybe pricing tickets to Scandinavia to get an award. But something else happened. A disruptive technology crashed the party around the same time. Okay, and this was disruptive, but also extremely good for cancer patients, and that's this field of immuno-oncology. People had been working on it for a long time, but it started succeeding, and the idea was to turn on the immune system to attack the tumor. And this was the clinical trial that really showed that it could work. This happened in 2011. This is an antibody against a protein called CTLA-4. I'll explain that a little bit later. The antibody is called ipilimumab or Yervoy or Ipi. And this is a Kaplan-Meier curve, if you haven't seen them. This is a plot of patient survival over time. Okay? And the normal way people used to measure patient survival over time is they'd look at the 50% cut point and sort of look at the width of that red bar that I have on the graph. That's the change in median survival. It's only a few months, 3-4 months. Doesn't seem that impressive. But in fact, no one had ever seen survival benefit, ever, in a controlled trial for metastatic melanoma. It's a terrible disease. But what's more interesting to everyone, and to me at least, is that blue bar, the area in that blue bar. Some patients are having long-term benefit, long-term survival from this therapy. You don't typically see that in chemotherapy. People are living 2 years, 3 years, over twice as many people are living 2 years and 3 years into this. And so, how is this working? Why is this happening? We've known literally for 50 years this year that if you have a tumor, your immune system has recognized it as foreign. You have antibodies against it. You have cytotoxic T cells against it, but yet you have a tumor. So this is actually an old version of the slides. So I'm gonna, in about 120 seconds, teach you immuno-oncology. So bear with me. You know, the standard way, biology 101, back in the 80s or even 70s, is we knew that there were antigen-presenting cells. They have this protein called MHC. They present foreign peptides, like peptides from a tumor, peptides that were floating around, grabbed by the APC and presented to the T cells via the T cell receptor. And when that engages, in the ideal world, it stimulates the T cells divide, and their children divide. You get a clone army that goes out and destroys a tumor. That's the idea, right? But often it doesn't work. At least by the time someone comes to a doctor, it hasn't worked. So the story is a little bit more complicated than this. It turns out that that signal is not sufficient. The T cell says, I'm not going to just turn on if you bind to my receptor. I want to see your license, your professional license, to give me this antigen. And that's a molecule called B7 on the antigen presenting cell. And when that's engaged, those two signals, that's called a co-stimulatory signal, is sufficient to turn on the T cell and get that T cell to divide. It's a clonal T cell that recognizes that specific antigen and can mount an immune response. And then it turns out that immediately after that happens, the T cell turns down that signal. It expresses a protein called CTLA4, which is the target of ipilimumab, and that outcompetes binding to B7, turns off CD28, attenuates the signal. And finally, there's another important pathway called PD-1, PD-L1. PD-L1 is expressed on the antigen presenting cell, PD-1 on the T cell, that also turns down the signal, okay? So the immune system is a very complicated adaptive system that seeks to balance activation and attenuation and reestablish homeostasis, okay? So that's immunology, immuno-oncology 101. It's more complicated than that. We've identified 14 different pairs of co-stimulatory or co-inhibitory molecules now, and everyone in the world is working on them because of the success of ipilimumab. Okay, and unfortunately, cancer, through the process of evolution and natural selection, or unnatural selection, has figured out how to use these pathways to its benefit. So many tumors actually express that protein called PD-L1 on their cell surface. It binds to PD-1 on the T cell and turns the T cell off. So it's a mechanism of resistance. All right, and the two main drugs that are available to treat patients today either target CTLA4, which blocks that co-stimulation event, or PD-1, PD-L1 antibodies that block that negative signal there. And so CTLA4 antibodies, ipilimumab, binds to CTLA4. PD-L1 blocks the negative signal from the APC or the negative signal from the tumor, and that's how immunotherapy works, okay? And that expression of PD-L1 on the tumor is actually called a biomarker, a predictive biomarker. For some tumor types, you can measure that expression and you can predict whether a patient will respond to PD-L1 therapy or not, or PD-1 therapy. And the data is getting better. This is a more recent clinical trial with IPI, with PD-1 antibodies, we call them NEVO, and with combination therapy. And you can't even measure the median survival here. It's a period of years, but it hasn't got to 50% yet. So over half of these patients are living a long time in melanoma, which is a disease that has a terrible prognosis. Okay, so we have the molecular targeted therapy of cancer, and we have all this immunology, all this homeostasis, co-stimulation, co-inhibition, and I was asked to try and find the connections between these two domains. If you're taking genetics and looking at some trait, like immune state, we call that a genome, and you do it genome-wide, we call it a genome-wide association analysis. The immune state, however we measure it, some continuous value, is called a quantitative trait in the study. And we end up building pretty simple linear models to do this association analysis. You have immune cells, maybe we counted the T cells on the left, and you want to know the association between that and any cancer mutation, any amplification or deletion of DNA, and plus we want to look across a bunch of different tumor types. Okay, and so we went to the TCGA database that I described and tried to use that data as the raw input to go into these models. And as I told you earlier, we have tumor mutations for 11,000 tumors. We have copy number amplifications copy number deletions, but they didn't measure or count immune cells in these tumors. So we had to figure out from those 7 different other data types how we were going to use that data to infer the count of different immune cells, the count of T cells, the count of B cells, the count of antigen-presenting cells. And so we ended up using the RNA expression from the tumor to do this. So the goal is to find specific markers for these antigen-presenting cells or T cells from the RNA samples of the bulk tumor and project those markers to estimate immune content. And the challenge is almost literally, you had a piece of tumor tissue that had blood vessels, that had tumors, that had tumor cells that had different types of immune cells, and you almost literally put them in the blender. I mean, it's analogous to a Jackson Pollock painting that's been reduced to like 1 gray pixel, right? So we ended up having to use some somewhat fancy correlation measures, not Pearson correlation, but other things using partial correlation theory and a metric that's very useful called mutual rank distances to try and deconvolute the RNA structure within tumors and try and recreate a painting. All right, and this is just an example of what a network reconstruction looks like from these data. This is all the 11,000 tumors in TCGA, and we pulled up some markers that we thought were good T cell markers, and we say, what are the neighbors and what's the relationship between the different markers? So on the upper left here, you have CD2 and CD3, which probably means nothing to you, but for an immunologist, those are well-known pan T cell markers, okay? And on this network diagram, they're also very, very close together in a co-expression network, which means they're probably coming from a single signal, they're probably coming from a single cell type, and we use those markers to estimate T cell content. On the bottom right, I haven't brought them up, but there's actually a category of bad T cells, suppressive T cells called Tregs, or regulatory T cells. And we were able to find 2 markers that, again, we believed were markers for regulatory T cells and also were very tightly co-regulated in a co-expression network. And we used those to project our estimate of regulatory T cells. So in all, we had about 10 different cell types that we could deconvolute this way. And that means we have on the left, we have our quantitative trait. Those 10 different cell types are estimate of content. So we throw them into an association model with cancer genetics, with tumor type and subtype, and we started to realize the data was incredibly biased. We had a problem. Nonsensical p-values, just everything was going in one direction. So the main covariate that we found was the fact that these tumors are coming in and getting their RNA sampled, their DNA sampled, but they were at various stages of purity, okay? And the less purity in terms of tumor cells, the less pure a sample is, the fewer mutations will be called, the lower the copy number changes will be estimated, okay? And this immune infiltrate is part of the impurity. So our data is biased, okay? And it's a bias embedded whenever you try and cross-compare tumor genetics with tumor RNA. And so we ended up building a purity covariate and adding that to the model. The second scientific challenge, and in normal human genetics, the people doing the work don't have this challenge. In normal human genetics, the genetics causes the trait. Your blonde hair does not cause cancer, it does not mutate your DNA, okay? In tumor biology, that doesn't have to be true, and I think we've seen at least a couple examples where the immune state, the phenotype, is actually causing the tumor to mutate and changing its DNA. So we don't have a cause and effect relationship we can determine by itself. Computation-wise, it was moderately expensive enough to make it difficult to debug. Sampling the data only gets you so far because the edge cases always are the ones that fail. And we also have a statistical challenge. Everyone in genomics has to work with this every day. It's a multiple testing challenge. I sort of think of it as a demon, as a multiple testing demon. Every hypothesis you make on the data takes away your statistical power. And we are making tens of thousands of hypotheses whenever you do a genome-wide analysis. So you have to get very strong signals for them to be significant. As sort of an aside, we intended to publish this work, and we ultimately did, and I went through the discipline of trying to write the paper in code, which I thought was a smart idea. It may have been a stupid idea, but was able to do it. So the way this worked is you start with R and Markdown code. The code downloads the entire TCGA data set, projects the immune cell estimates, does the association study. That goes to knitr, which creates an integrated text table and figure report. And that goes to pandoc, which creates a publication-ready Word document with publication-ready texts, tables, and figures, and also was able to integrate the bibliography from multiple different sources. All right, I'm going to spend the last few minutes going through a couple of the high sort of top-line results from this association study. And the first one I want to talk about is a deletion of part of chromosome 9. It's called 9p. You look at chromosome 9, and in that center graph, you have a normal chromosome 9 on the left and a chromosome that has a part of 9p deleted. You can see it's smaller. A piece has been cut out by the tumor. And when tumors have done this on both copies of 9p, that graph on the right side is a normal cell with 2 copies of 9p, and on the left is a homozygous deletion. That tumor has cut out both copies of 9p. And our estimates of immune cells are dramatically lower in those tumors. Those tumors are immune cold, okay? And we wanted to know or understand why. So what you can do is you can go to that region of 9p, see what's there, what's being cut out. And what we found is something I brought up before, this cell cycle control gene, CDKN2A. That turns out to be what the tumor is really trying to get rid of. It wants to delete CDKN2A, remove cell cycle control. So this is the driver event for the loss of 9p in tumors. And then you can say, well, let's look on the map here and what else is around. Interferons. Interferons you've probably heard of. They're stimulants for the immune system. They're part of the innate immunological system. We didn't find 1, we didn't find 4. We found 17. This is the largest cluster of interferons in the human genome. They're sitting next to CDKN2A, and they're getting deleted a lot when CDKN2A is getting deleted on chromosome 9. So our current view is that this is a collateral damage event in cancer genetics. The cells are deleting CDKN2A in order to grow. But while they're doing that, they've deleted a very strong component of the immune system within the tumor cells, and that's what's making these tumors cold. The second case I want to talk about sort of goes the opposite way. So this is a mutation on chromosome 8. I put a little star next to where this gene called caspase 8 is. Caspases are components of the programmed cell death pathway, which is built into every cell that we have. These are signals and effectors that tell the cell and help the cell to actually die, force it to die, okay? And they're used by the immune system to trigger cell death from T-cell attack. And the tumors that have these mutations actually have a lot more infiltrate than the tumors that are normal wild-type caspase 8. And so our current interpretation of this is that this is one of those adaptations where the immune system has infiltrated the tumor, it's trying to kill the tumor cells, and the tumor cells are mutating caspase 8 to avoid cell death and to escape. So the obvious question is this going to relate to therapeutic response? These genetic changes, there's probably 50 interesting hypotheses from these papers, and you have to, from this association study, and you have to follow them up. We have at least anecdotal evidence from the literature, from Pam Sharma's group, that patients in melanoma that have lost this region of 9p in a small clinical study, none of those patients responded to immunotherapy. But again, this study was small. We need more work. Finally, I want to talk about in ways a simpler marker as a proxy for tumor genetics and tumor mutation. What if instead of looking at all these individual mutations, assessing their association with the immune system, what if you just sequence a tumor from a patient, count the mutations, and use that as a number? Total mutational burden, just count them up. Okay, and this is data, another survival curve, or pair of survival curves. It's a post hoc analysis from a trial we did in lung cancer, and that trial did not achieve its endpoints. And at the time, we didn't have the tumor mutation data from all of these patients, but we went on post hoc and acquired it, and the important lines are the two blue lines on the upper and lower survival curves here. So down at the bottom, those are tumors that had many mutations, that had a high tumor mutational burden. And up at the top are the survival curves of the patients that had few mutations. And I think you can see there's a pretty dramatic difference. And so this TMB, this tumor mutational burden, is emerging as a second biomarker used to predict which patients are going to respond and which patients are not in immunotherapy, in addition to the measurement of PD-L1 on the tumor that I talked about earlier. Okay, I'll summarize. I tried to take you through two really, it's an exciting time to be doing what I'm doing in biology and in cancer genomics. We have revolutions in the targeted therapy of cancer and in immuno-oncology. The people doing this work early on were from very different backgrounds, which is why the work seemed to be coming from two different planets with not too much known about crosstalk. But my company, myself, many other research groups both in industry and academia are trying to create models that unify this work and understand which patients are going to respond, particularly to the immunotherapeutic agents. Okay, and a very, very thank you and a very, very profound thanks to the patients and their families that enroll in clinical trials. They're sick, they get poked and prodded, but their contributions are critical to every part of the work that we do. So I have 2 minutes. Thank you.
I'm really impressed by the level of statistical sophistication that this analysis has showed. But one of the things that I found was kind of interesting is in some of the reading that I've done, it's been suggesting that the cost of drug and therapy development has been growing at a super linear rate. When shown against that graph of the cost of genetic sequencing, I think that that's really interesting. Is it just that the low-hanging fruit is picked and things are a lot harder now than they were in the past, or?
That's a complicated question. So it's quite clear that, and it's not just BMS, there are multiple companies now with agents against PD-1, PD-L1, CTLA-4, and every company is working on all the rest, okay? But really, the CTLA-4 and the PD-1 agents, particularly PD-1, seem to be the most dramatic. There are 22 indications for which these therapies are approved now, starting from 1 in 2011. And they're expensive, but the interesting thing about them and the exciting thing for me is that it's actually extending survival. I didn't talk through it, but at least in melanoma, the patients, if you have survived 3 years, it looks like the survival curve plateaus. Okay, so this is incredible benefit for patients. So they may die of something else.